Alright, so today we're going to be looking at Gregor Mendel, the quote, father of genetics, and monohybrid crosses or Punnett squares. So with that, Mendel first started looking at genetics through pea plants, like the peas you eat, your vegetables, and found that, what we know now, that organisms have two alleles for each trait, one from mom, one from dad. Okay. The dominant trait is always shown. We know that. We call that the law of dominance. That was from him. And then the two alleles for each trait will separate whenever meiosis happens, and segregation was the word he used, and so he calls it the law of segregation, which means they separate and there's a chance you get one or the other. Alleles for each trait, they don't relate to any other trait. We call it independent assortment, which means just because you have blue eyes doesn't mean you have blonde hair. Sometimes they go together, but you can get one or the other. It doesn't matter. They don't have to be one for one. It doesn't have to be if you have blonde hair, you're going to have blue eyes. All right, that's the law of independent assortment. Okay. All right, so Punnett squares. This is showing those three laws that Mendel came up with all together in one place. Okay, we use these squares to predict what's going to happen. My pen here isn't working. All right, so used to predict offspring of a cross. And we have four squares. This is if you're looking at one trait. Okay. Alright, so if we're looking at one single trait, we have a four square Punnett square. Alright, so this is for one trait. We call that a mono, which means one, hybrid cross. Hybrid meaning something different. So one trait different from each parent. Okay. First you must look at the gametes. These are the sex cells. So a parent that has the genotype big B, the only thing they can pass on to their offspring is a big B. Those are the gametes that are possible. A parent who has little b can only pass on a little b to their offspring, because that's the only possible thing they have for their two gametes. All right. A parent that has the heterozygous big B, little b, has a 50-50 chance of giving a big B, a 50-50 chance of giving a little b. You don't know. It's whatever that egg or sperm has. That's what you get. You don't know what you're going to get. Okay. So, example here. Independent assortment means that these two alleles for each parent are going to separate. That's the law of segregation. All right, they're going to separate into the two different gametes. One of these parents gets written on the left-hand side of the Punnett square. The other parent gets written on the top. So I take one gamete, big B, and I put it on the left. Sorry for the writing here. Trying to do it with a mouse. I take the other allele and I put it in by the next square. That's this parent. For this parent, I take the first allele and I put it on the top of the first box. I take the second allele and I put it on the second box. So now this allele is going to be in every square in that column. This one, every square in that column. And this goes with the rows. So big B would be written here from this parent. Big B from this parent. Do the same for the next one. Bring this big B over. Big B. Bring this little b down. little b. Now, why did I write the big B first? Because if it's dominant, it's going to show that's what we write first. Anytime you have a dominant allele, it gets written first. Okay? We do the same thing here. This is a big B, so it gets written first. And little b. And then this last box, we have little and little. Doesn't matter which one you write first because they're the same. Okay? So now we have our 4 possible offspring for these 2 parents. Alright? So, what we look at is the next slide. That's what we have. Okay? Big B, blah, 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 blah. Now, if we're looking at possible genotypes for these offspring, we group them. We have this one, big B, big B. So we have one of those. Total number of squares, we have 4 squares. If we have 1 square that's big B, so 1 out of 4 is 25%. So 25% big B. Sorry for my writing, this is bad with a mouse. Alright, that takes care of this one. These 2 are the same. So we have 2 out of 4, 50%. That is, just going to write 50, you know it's percent, 50% heterozygous. Alright, and then we have our last one here. It's 1 box, 1 out of 4 is 25%. So 25% big B, or excuse me, little b. That's for genotype. Right? The possible genes that are going to be given to the offspring. The phenotype. Remember, phenotype is physical characteristics. Well, we can't do that. We can't do it for this example because they did not define what is dominant to what and what trait this is. Okay? So if they would normally in the problem, they would tell you black hair is dominant to white hair, and we'd use that for the phenotype. Alright? But at the same time, I do know that this one has at least 1 big B, 1 big B, 1 big B. So these 3 would show the dominant trait. That would be 75% would show the dominant. And then 25% would show the recessive. So that's the only way we get recessive is if we have both littles. Okay. Making sense so far? What we want to look at is how these traits independently assort, how they segregate, and then how they, what's the possible offspring. That's what Punnett squares do. It predicts the possible offspring that 2 parents will have for that certain trait. Remember, we're only looking at 1 trait with this Punnett square. It's just 1 single trait. We will do some practice with this tomorrow. There will be a worksheet that you have to do. I am open to some emails if you have questions. However, I'm not going to get right back to you because I'm going to be teaching class. But at the same time, if you do have questions, this stuff can get difficult. Going into Friday, we're going to do dihybrid crosses. Dihybrid means 2 traits that we're going to do. And it's going to be more. It's going to be more squares, more things involved. All right? That is what we have for today. Go do your drill, and you are caught up for the day.